So today I will discuss how invariant feature selectivity is achieved in cortex, and in particular I will describe a trade-off between complexity of sensory representations and invariance. And I hope that this observation is related to the ideas that we heard from control theory, and also the relationship between the sensory representations and control theory. So why is this an important question? Well, it's invariance and selectivity together form, it's easy to be either invariant or selective, but together this forms a cornerstone of our ability to recognize objects. And as many of you know, this is a very difficult problem because we cannot do it just by template matching in terms of pixels for a given object. So here I'm taking one photograph of a Apollo statue, and different people on different days take the same different photographs. So the same object, but when viewed from slightly different angles, generates a range of images, and essentially we never see the same image twice. So the question is, how can we recognize a particular object, especially that if I take another object for a given point of view will be very similar, but of course is different. So I would argue that the question of feature selectivity and visual perception is probably, one of the, aside from the motor representation, is probably one of the main things that the brain does, because there are general principles that generalize between vision and auditory and other senses, but even if we look at vision, so this is an example from a classical paper by Fellman and Van Essen, where they colored all areas that participate in the visual object recognition. So we know that it's approximately between 1/3 and 1/2 of the primate brain, plus if we add other senses. So if we understand even visual object recognition, we will go a long way towards understanding what does the brain do. So the previous talk in this session already alluded to some prior knowledge that we know how the visual processing in the brain occurs, and in particular I will also show this an example, of course, from the classical paper by Hubel and Wiesel, which shows that, so this is in the primary visual cortex in the back of the head. One can find neurons such as, this is from their paper, where neural response is measured by the number of spikes, and for that particular neuron, if they presented an orientation that was close to horizontal, there will be no spikes, and for as the orientation approaches, for example, 45-degree line, there are more spikes. So this is just one neuron, but a body of work over the course of many decades argued a picture that in the primary visual cortex, or V1, we take the visual image and we represent it in terms of edges. Well, it's simplifying, but to the first approximation, that's what V1 does. And then we know that at the end of the visual processing, we find other neurons. So this is the data from DeSimone and colleagues. So they show another neuron, so in this case, a neuron that is selective for a hand. And we know that it's selective for a hand because it produces lots of spikes. If you simplify a hand to a mitten, then you get fewer spikes. This particular neuron was not selective for a face, but if you show a hand at 90 degrees, the response comes back. So that illustrates the question of invariance and selectivity. I could say that in order to build a detector like so, I will integrate all edges that form a hand. But then I have to also integrate across all positions. And then I have to integrate across all orientations. And then across over all scales, if I would like to recognize a person as they walk towards me along the corridor. And at the same time, so that argues for a large degree of convergence. And there are studies that show that within, if we look at the middle area here V4 that I will be focusing on here, essentially up to 1/6 of this huge V1 surface projects to a single V4 neuron. So the degree of convergence is enormous. If this convergence was without any principles, we would have lost all selectivity. So there must be some principle according to which this convergence is organized in order to preserve the selectivity. So what I have been trying to do for the past several years is to understand the intermediate steps of the transformations that can help us go from detailed representations in terms of edges into representations that are highly selective for complex objects and invariance. So to address this question, we will be analyzing, today we'll describe analysis of neural responses in this intermediate area V4. And, but before I do that, I would like to begin with the theory and quote some results from the computer vision. So in computer vision, of course, the edge selectivity is one of the main steps of almost any recognition algorithm. But then how to build invariance, and so here I'm quoting the results from Ullman and Solovyov, and they ask, suppose I would like to build a detector that is a partial letter A that is invariant for position. So we start with an image where this letter can be at various positions. And unless we would like a detector that is specifically for the shape, we will start with little segments. And we can imagine that we will start combining these segments in building more and more complex patterns. But the question is how quickly we should increase the invariance. For example, this little feature here that corresponds to a little tail. So it will be invariant right away. And these little segments, they will be partially invariant. So the idea is that in order to preserve, the qualitative idea is that in order to preserve feature selectivity and invariance, I have to build up invariance, but in such a way as to avoid potential confusion. So if I just say that I want this invariant feature and this invariant feature, then instead of being selective for a particular angle, I will also be responding to their crosses. So we would like to build detectors that are small, but in the range of invariance will still be tolerant towards, will minimize misidentifications. So qualitatively, it leads to two observations. One is we have to gradually increase complexity of features and gradually increase invariance. And potentially we can expect to see, and also the invariant of these features, so then there will be one complex feature, so it has to be interleaved, the integration of invariant representations and feature complexity. But also we can expect to find a trade-off between complexity of features and the degree of invariance, and that's actually what we found. I think it relates a little bit to the control questions that I will finish at the discussion point. So, as a preview, in order to analyze data like this, we started to develop methods where we can take neural responses to natural stimuli. So there will be on the order of 20,000 different images, sometimes dynamical images. And then I will look for example, one feature that together with position invariance will account for the responses of a neuron. So we can fit this model with maximum likelihood to a given data set, and then test predictions on a different part of the data set. So again, parameters, so we begin with a statistical data set of large number of stimuli that were presented, large number of responses. Typically, 20,000 of stimuli will give me several thousand of responses, and from this I can build a feature in a grid of, say, 100 points. So each time we lose an order of magnitude. And parameters of the model will be what are these profiles that are relevant, what are the features that are relevant for the response of this neuron, and the range of, say, position invariance. In this model, framework, of course, generalizes for other types of position, other types of invariance, but we have to always, at least in this case, assume that if it is scale invariance, position invariance, rotation invariance is more complicated. So we have taken data, was taken in collaboration with laboratory of John Reynolds at Salk Institute. And as I mentioned, we have analyzed responses of awake animals as they were watching 3-second long movies. There were between 1,000 and 2,000 of these individual movie segments that were concatenated into a data set in order to analyze the neural responses. So here is what we found. First, we found that adding position invariance improved predictive value of the model on a novel piece of data. So I characterize the predictive power in terms of information explained, but we can use other measures as well. So what's important on this graph is that predictive power of models with position invariance is essentially always greater than models without position invariance. And that's an argument for, yes, the sort of the global position invariance is important for these before neurons, meaning that if I take one template and allow and move it around, that improves the predictive aspects, that better accounts for the V4 responses. By comparison, if I impose scale invariance, that decreases the predictive power of the model. So for now on, I will be focusing on position invariance. So in the presence of position invariance, what were the features that accounted for the responses of these neurons? So here is one example of a feature. It's shown as a piece of a segment as a function of horizontal position and vertical position. And while it is a complex shape, we can fit it in terms of what I called a curved Gabor model, as in if in V1 a segment would be straight segment with a Gaussian envelope. So here we have the same model but along a curved contour. So in this way we can summarize the feature selectivity of a given neuron. I acknowledge that maybe there are more than curvature to these neurons, but this is the first pass through a data set. And even though none of these stimuli were presented in the experiment, but when we find evidence of curvature selectivity, it certainly echoes prior work in this area by a number of groups where people have supposed that perhaps these neurons would be selective for curvature. One can sort of guess that if V1 is selective for straight edges, then the next element in complexity would be either combinations of lines of different orientations or a smooth curve. So while in prior work they explicitly probed curvature selectivity, here we get it without a prior assumptions. But we can go further and ask whether there are multiple features that at a given position affected the neural responses. So I'm potentially missing a slide, but The idea is that in addition to accounting for position invariance at one, looking for one feature, we try to estimate what were the combination of features at the same position that accounted for null responses through some nonlinear function, and then allow a global position invariance. So there is in this case, 2 features per position that we would like to estimate. We allow for arbitrary nonlinearity within position, and then allow for integration across different positions. So what were these 2 features? So this is the result of real neurons. And for 2v4 neurons, this is the first feature, so the maximally informative feature, and then the second feature, a second maximal informative feature. And this is the result of this curved Gabor fit to the pattern that was reconstructed from neural responses to natural stimuli. So this is again neuron 1, one column, neuron 2, another column. So in both neurons, we find that, yes, indeed, there is a curved pattern, and typically the curved pattern was similar for the same neuron. So the 2 features coordinate to have the same orientation and curvature. But what is different between them is, for example, here you can see that there is a negative region flanked by 2 positive regions. In contrast, for the first feature, we see 1 negative region and 1 positive region. So for those that work on visual processing, this type of selectivity is certainly reminiscent of a canonical model of what happens in the primary visual cortex. So in the primary visual cortex, when I said that neurons are selective for edges, that was a simplification. And in particular, there are different types of cells, and there are simple cells and complex cells. So for complex cells, their description, their minimal description, is in terms of 2 features. And they are select, for example, it would be selected for an edge, but it would be 2 features. They will have the same orientation, but one of them will be an edge, and the other one would be more like a bar. So in terms of, if we think about a fitting Gaussian function with multiplying an oscillating function, then one of them would be a cosine in cross-section perpendicular to this preferred orientation, and the other function would be a sine. So why is this important? Well, it is important because if I take edges at different position, slightly displaced edges at different position, then the difference between them, the difference between 2 edges will give me a bar. So with this combination of these 2 features, I can account for local position invariance. So this can be better illustrated by going back to the classical scheme that Hubel and Wiesel proposed, where they said, well, retinal neurons feed into simple cells that are selected for an edge. And these simple cells, there will be a multiple set of simple cells, each selected for an edge. And as a result, we will get this description of a complex cell that I talked about, which will be in terms of the this pair of sine and cosine. And once we observe this feature selectivity, which in terms of the sine and cosine, it's called a quadrature pair, meaning that these the 2 Gabor features have all the same parameter except for this phase of the oscillation. And that's the model of the complex cell. So now what we see is a very similar picture, but in V4, and not with respect to a straight contour, but with respect to a curved contour. And to my mind, it suggests a simple scheme of how signals from V1 could be combined in order to preserve both local position invariance and global position invariance. So every time we see a quadrature pair selectivity, I take it as an indication that there's a presence of local position invariance. And then we also have, And then we also have global position invariance, which was part of the model when I estimated which two features together accounted for null responses in the presence of global position invariance. So we have multiple types of position invariance. So after showing you example neurons, I'm showing you some population data where each point is a pair of features for real V4 neurons, and I'm plotting the orientation of feature 1 versus orientation of feature 2 to show that they're correlated. And this is, of course, reminiscent of what we expect for normal V1-like, so quadrature pair selectivity, where the two features have the same orientation. So in V4, we get these features that have similar orientation, similar curvature, but different spatial phase, meaning that the cross-section of the oscillations in the direction perpendicular to the contour. And I'm plotting here across the distribution this spatial phase across the distribution. And for a canonical quadrature pair selectivity, we expect here a sine and cosines, that is a phase difference of 90 degrees. In V4, we find a peak of 60 degrees, but I think this is because versus these two features, they not only have a spatial phase difference of 90 degrees, but they're also orthogonal. If I take a purely 90-degree spatial phase difference along the curve contour, these two features will not be orthogonal. And because for technical reasons, these two features are orthogonal, and so that induces a phase shift away from 90 degrees. So what I showed so far is that there is this quadrature pairing, and this is across now not 2 neurons, but 6 different neurons, and where I'm showing first features in the first row, second features in the second row, and 6 different neurons. And what you can appreciate is that these neurons differ in the degree to which their preferred curvature exists. So this one is a more curved, and this one is more shallow. And now another part of the analysis comes out. It turns out that the range of the global position invariance correlates with how curved these features were. That is, the range of global position invariance increases from left to right. And across the population, this is the result of the analysis where each dot is a pair of features representing one neuron. And on the left, on the x-axis, I'm plotting the range of estimated range of position invariance. And on the y-axis, I plot the estimated curvature selectivity in relative units with tighter curvatures on the y-axis, on the high end of the axis, and more shallow curvatures on the bottom axis. And we see the inverse, statistically significant inverse relationship between curvature and invariance, meaning that neurons that are tuned for tighter curvatures are less position invariant than neurons that are tuned for shallow curvatures. So this was a prediction obtained using natural stimuli, but usually people ask how well does it generalize to parametric stimuli. And to address this concern, Anirvan Nandi was a joint postdoc, and he did a special experiment with probing these V4 responses with little curved segments that had 3 pieces, 3 segments, and in this way one can approximate different curvatures. And actually what they found was the same phenomenon, such that neurons that had larger position invariance had shallow curvature. So here is one example of what were the preferred segments, line segments for this neuron, and plotted are all segments that had more than 95% of the peak response. And for this neuron, most of the features have the same orientation. In contrast, for another example, V4 neurons, they all have different orientations, and the curvature of each feature is greater. So one can also plot it across population to show that the range of invariance with more spatial invariant index on the y-axis versus curvature selectivity on the x-axis, there is an inverse relationship, similar to what we have found with natural stimuli. So far, to summarize the findings in visual area V4, we find that position invariant models yield improved predictive power, and that's argument for more global position invariance. And then in addition to global position invariance, the models indicate the presence of local position invariance, which we find because of the selectivity of a certain type of selectivity to two predominant features that had the same curvature, same orientation, but different spatial phases. And and also there is a trade-off between curvature tuning and position invariance. I would say that the motivation is that if I'm trying to perceive a contour that perceived an object, I would like to know the position of more curved segment to a greater precision than position of more shallow curvatures. So there are a number of functional reasons for why that trade-off might be useful. And so last and not least, I would like to acknowledge people with whom I had the pleasure of working. The V4 work was done in the laboratory of John Reynolds. Jude Mitchell generated synthetic stimuli, and Anirman Nandi recorded data from synthetic stimuli. Minjun Ko recorded data from natural scenes, and two students, Ryan Robinkamp and Michael Eichenberg, worked on the methods that made possible these analyses. And so the work that I presented today is described in these publications. The methods paper is in neural computation. The trade-off and quadrature pair selectivity was published in PNAS. And in my recent review earlier this year, I also offer some parallels with auditory processing, which I didn't have time to talk today. Thank you for your attention.